Okay, let's unpack this. Well, forever, we've looked at creatures like salamanders, haven't we?
Absolutely. Just regrowing entire limbs like it's nothing. It's pretty amazing.
Well, we mammals, including us, we've always thought we're pretty limited on that front. Regeneration just isn't our thing. Or is it?
Well, that's the question, isn't it? What if our own cells have maybe a hidden capacity, something that challenges that old assumption?
Right. And today we're taking a deep dive into exactly that. Specifically, how our cells react when oxygen gets low. That's hypoxia.
And then there's this really crucial protein involved, hypoxia-inducible factor 1 alpha, HIF-1α.
HIF-1, okay. And it's a transcription factor, so it controls genes.
Exactly. Turns them on, turns them off. But normally under regular oxygen conditions, it's degraded almost immediately. It just doesn't stick around.
Okay. But the really interesting part happens when oxygen levels drop, like after an injury.
Precisely. Suddenly, it doesn't get broken down. It stabilizes, builds up inside the cell.
And once it's stable, it gets to work.
Yes. It activates a whole bunch of genes. Crucially, ones needed for angiogenesis that's building new blood vessels.
Through that VEGF-VEGFR2 pathway you hear about.
That's the main one, yeah. Critical for bringing in oxygen and nutrients. Plus, genes for metabolism, keeping cells alive. It's a whole survival package.
So our mission today is really to explore this HIF-1 pathway. Can we maybe tweak it? Enhance our own repair mechanisms?
Potentially unlock abilities we didn't think we had. That's the exciting prospect.
It's quite elegant, really, this oxygen sensor.
It is. Think of it. HIF-1 is always being made, but enzymes immediately tag it for destruction when oxygen is plentiful, like a super-sensitive switch kept off.
So it's always there, lurking, ready to respond if needed, but usually the body keeps a tight lid on it.
Exactly. But injury, low oxygen, that degradation system just stops cold.
And HIF-1 builds up, like a distress signal going out.
And then it activates those genes, especially for angiogenesis via VEGF, VEGFR2, trying to fix the oxygen supply problem.
It really makes you think, doesn't it? If the capability is there, can we sort of trigger it deliberately, even without the usual low oxygen injury trigger?
That's where the research is heading. And yes, labs are doing just that. Finding ways to artificially stabilize HIF-1.
With drugs or maybe gene tricks.
Both, yeah. And the results in models like diabetic mice, they usually heal quite poorly.
Right, that's a big problem.
But if you stabilize their HIF-1, their wound healing improves dramatically. Significantly better outcomes.
Wow. Just from boosting this one factor, that's quite something. But what about real regeneration, like the salamander limb stuff? Is HIF-1 involved there too?
It seems to be a key piece, yes. People are looking at how HIF-1 affects epigenetics.
Epigenetics, like how genes are used, not the DNA code itself.
Exactly, like chemical tags in the DNA or related molecules, specifically a modification called MERA-methyladenosine.
Okay, MERA. Why is that important?
Well, in amphibians, this MERA process is absolutely vital for forming a blastema.
The blastema. That's the blob of cells that actually does the regrowing.
That's the one. A mass of undifferentiated cells ready to rebuild complex tissue, and Mohel helps make it happen.
Okay, so are we seeing anything like that in mammals when HIF-1 is active?
We are starting to see things described as blastema-like structures, even in mammals.
Seriously, blastema-like?
Yeah, particularly when HIF-1 is activated during injury repair. Now, it's not full limb regeneration yet, not by a long shot. This is early days, lab research.
Still, the idea that mammals might have that latent potential, it's fascinating.
It really challenges that old dogma that mammals are just non-regenerative, full stop. The pathways are complex, but the hints are there.
So wrapping this up, what does this all mean for you listening? This dive into HIF-1, it really shifts things, doesn't it?
Definitely. We see artificially stabilizing it, helps wounds heal, even in tough cases like diabetes models.
And the link to epigenetics, that MEIS modification, which is so key for amphibian blastemas.
And now seeing those blastema-like structures potentially appearing in mammals when HIF-1 is active, it just opens up so many questions.
Yeah, it challenges that core belief that we mammals just can't regenerate complex parts if our bodies have this latent ability, this potential.
What else might be waiting to be uncovered? What other biological tricks do we have up our sleeve?
Makes you think, right? If we can learn to activate the right molecular cues, like this hypoxia response with HIF-1.
Could it completely change how we approach healing, recovery, maybe even aging someday?
Yeah.
It's a space to watch.